and what is the outer part of the steel? Epidermis, steel. What is the remaining part? Hello?
Sir,
What is remaining part? You tell me, that is correct.
pericycle, sir.
No,
Endodermis,
pericycle is,
endodermis and pericycle.
no, actually, what is the complete region? In internally, three clear regions are there. What the three regions are? First one, epidermis, outer,
Epidermis, cortex and steel.
right. Now this is still completed. Epidermis completed. What is the remaining part?
Cortex, sir.
Cortex. This is cortex. Cortex. In cortex cells also change as a meristematic tissue and form the cork cambium ring. Cork cambium. This is called cork cambium. In cortex cells, cork cambium in cortex cell, vascular cambium inside the steel. Now, due to with help of this cork cambium and vascular cambium, secondary growth takes place. Is it clear first? Hello?
Yes, sir. Yes, sir.
Right. So let's now let's see the formation cambium ring. Before formation cambium ring, that is already what are the requirement is the lateral meristems, that is vascular cambium and cork cambium. Both belongs to lateral meristem. First vascular cambium ring formation or next one cork cambium formation. Before that, in secondary growth, there are the changes occurring during the secondary growth of dicot stem can be divided into two stages. They are intrastellar changes and extrastellar changes. First, see intrastellar changes. Intrastellar means inside the steel. Now here, uh, intrastellar. First, intra steel. This is first step. What happened? Let's see the intrastellar region. Formation of cambium ring, first one. Let's see how the cambium ring are formed. Listen carefully. In dicostem, the cells of cambium present between the primary xylem and primary phloem, that is called intra. That is called intravascular cambium. The cells of medullary cells adjoining this intravascular cambium become meristematic and form the intervascular cambium. This both together form as a cambium ring. Cambium ring. This is cambium ring. Now cambium ring formed by intra and inter, both vascular cambium. The first, let's see the formation of cambium ring. After that, let's see the activity of cambium ring. As I told just now, uh, are you able to see the diagram? Hello?
Yes, sir.
Right. Inside the steel, the vascular bundle. Are you able to see the vascular bundles also?
Yes, sir.
So these are the vascular bundle, right? Inside the vascular bundle, this cambium called as intra intra intravascular intravascular and between the vascular bundle empty space. Are you observing the empty place? Hello?
Yes, sir. Yes, sir.
What are present in this empty place? Actually, this empty space present with medullary rays, which rays are arising from the medulla, center medulla. From the medulla, medullary rays are arised. This like this medullary rays are arised. Later, what happened? These medullary rays change as a meristematic cells. Change as a meristematic cells. Meristematic cells. Now it is called as a meristematic tissue. Now where is the ah? What is? Tell me. And marker is clear or not? Hello?
Sir,
Your voice not clear. Please tell again. Hello?
sir,
Yes.
Any disturbance from my side?
not your side, sir.
Okay. So far clear?
Yes, sir.
Right. So this medullary race actually convert into meristematic tissue. Now this meristematic tissue between the vascular bundle meristematic tissue, this is called interfascicular. Interfascicular. Interfascicular cambium. Interfascicular. Intra and inter, the both together form as a ring. This is cambial ring. This is, you can see, this is interfascicular cambium. This is intrafascicular cambium. Now this is completely called as, now this ring, this entire ring called as, this ring called as cambial ring. Now clear, cambium ring formation clear? Hello?
Yes, sir. Yes, sir.
Any doubt in this?
No, sir.
Right. So intra, which is present inside the vascular bundle, inter, which is present between the vascular bundle. Okay?
Okay, sir.
Right. After formation cambium ring, what happened? Let's see. This what important. Cambium ring, intrafascicular. Now let's see the activity of the cambium. After formation cambium ring, what happened? After formation cambium ring, the cambium ring active and begins the cut off new cells both sides, inner and outer, inner side and outer side. For example, uh, already somehow this cambial ring developed. This is cambial ring, right? After formation cambium ring, what happened? The cut off new cells, inner side and outer side. Inner side some cells, this side, outer sides, let's change color. Outer sides also formation takes place. The cells cut off toward the pith matured into secondary xylem, this side, secondary xylem, towards pith side, this medullary side, this medullary pith inside already. Just imagine like this. First of all, vascular bundle. Already in vascular bundle, primary xylem present. This is primary xylem. This is primary phloem. But after formation cambium ring, what happened? This secondary xylem also formed. Secondary xylem cells, secondary xylem. This is the secondary xylem. What happened during formation secondary xylem? The primary xylem goes toward the center. Isn't it? Hello?
Yes, sir.
The primary xylem already existed in primary structure, but after cambium ring formation, due to activity of this cambium ring, both the xylems are formed inside and outside. The inner side toward the pith, secondary xylem formed, outer side secondary phloem formed, right? So here can clearly given the cut off the toward the pith or medulla mature into secondary xylem, and the cell cut off toward the periphery outside, secondary phloem formed. So now the cambium, generally more active the inner side, more active inner side. That why here the formation of secondary xylem is more than secondary phloem. This is primary xylem. Primary xylem. Now this is secondary red color secondary xylem. It occupies the more space. Outside already, oh, sorry. Ring. I've taken only one vascular bundle. Just imagine remaining one. Already primary xylem present. Primary phloem present. But inner side formation, more xylem. More xylem. But outer side, very less phloem. This is phloem cells. Secondary phloem. The green color, secondary phloem. SP పెడుతున్నా. Secondary phloem. So in this activity, the secondary xylem is more than secondary phloem. Right? Is it clear? Hello?
Yes, sir.
Right. So secondary xylem produced is more. Secondary xylem is produced is more than secondary phloem and soon forms compact mass. So this total called as the some mass like structure like this is completely secondary phloem and this is this inner side completely it change color secondary xylem. It's completely secondary xylem. What happened during formation secondary xylem? The medullary region gradually decrease or increase? Hello?
Decreases.
Right, it's completely decreases and this place replaced by primary xylem. Later, the primary xylem also destroyed inside this medullary region. Right, is this activity clear? Hello?
Yes, sir. Yes, sir.
Right. So what happened during secondary tissues increasing inside and outside? Any any effect on the epidermis? Is there any effect on epidermis due to the increase of the secondary tissues?
A pressure will be formed.
Right, pressure will be formed on epidermis. The exact pressure will form on the outer external region. Due to the external region, this external region also need to be increased. This external region also need to be increased. That's why in external region also the external cambium ring formed. Like the stellar cambium, the external cambium also formed. That is called extrastellar changes. Let's see the extrastellar changes. First intrastellar clear. Hello?
Yes, yes, sir.
Right, right. And what happened during first let me completely about interstellar. Later we will go to the extrastellar changes. Right. But what happened here? The primary and secondary phloem get gradually crushed due to the continued formation accumulation of secondary xylem. Already told you. Now the primary xylem, however, remains more or less intact around the center. And at the same at some places, the cambium forms a narrow band of parenchyma which passes through the secondary xylem and the secondary phloem in the radial direction. These are the called secondary medullary rays. That means apart from the let's try again. Cambium ring, vascular bundle, vascular bundle, vascular bundle. So where the vascular bundle present? In the region of the vascular bundle, this region only formation secondary xylem and phloem. Secondary xylem inner side, secondary xylem inner side, and secondary phloem outside. But in the cambium ring, the between the vascular bundle inside the cambium ring, here this region, secondary medullary rays formed at both sides. Secondary medullary rays, secondary medullary rays formed both side, inside and outside. Right? Apart from the secondary xylem and phloem, these secondary medullary rays also formed on both sides. Both sides. And let's see the how the formation growth rings are annual rings. Growth rings, annual rings. After that, heartwood and sapwood. So actually, growth rings are annual rings formation. The first, the mainly which is formed in the temperate region or cold region due to the activity of the cambium ring. Just now we have seen the activity of the cambium ring. This activity of cambium ring actually influenced by the seasonal variation. During the favorable season, that is from spring season, what happened during spring season? The same cambial ring here, same cambium ring, one cambium ring, spring, when the came during the favorable condition, that is spring season, when more leaves and flowers are formed, right? Now, when the formation more leaves and more forms, what about the requirement of the water and minerals? The requirement of water minerals are
More water.
large or less,
It need more water.
more water. That ends the wood formed in this period shows more number of xylem vessels, having wider lumens. This is known as spring wood or early wood. That means, for example, now this is spring season. Now the secondary xylem cells are formed very. The secondary xylem cells are formed very highest amount because the requirement of the water minerals very high during formation of leaves. What is the spring season? In spring season, newly formed flowers and leaves. That's why the requirement of the water minerals high. That's why here the xylem cells also formation abundantly formed. Right? So this would call as now this time would form as uh, called as this is springwood. This is called springwood because it's formed as a spring season, called as spring season. So one of the ring completed, for example, here. Right. Now during the unfavorable condition that is the autumn season. Autumn. In autumn season, the plants are very less active and do not require more water minerals because this is the in this autumn season what happened during the flowers and leaves mostly they are fall off. That's why the when the flowers and leaves the fall off, any requirement of water and minerals, hello,
Less water it needs.
less water, and the food wood produced in the period show less number of xylem vessels having narrow lumens when compared with the spring. This known as autumn. That means the same. This is one ring form. The latter, for example, here if the continuation this ring, simple, right. I have drawn one side only. This is cambium ring. First, let's go red color. I have drawn only inner side, but not outer. Cambium ring. For example, first in spring season, more xylem cells formed. More xylem cells formed. In autumn, less xylem cells formed. Let's change color. Less xylem cells formed. This is blue color, less. Autumn. Autumn season. Spring season. Or autumn wood are called as late wood. Spring wood are called early wood. In this way, two types of secondary xylems or secondary xylem, another name is wood. Don't get confused with both words. The wood are produced in the 1 year. So this appear in the form of circle. The alternately in the transfer section, if you see the mature tree trunk, there is growth rings are formed. Now this is one ring. This is one ring for 1 year. Like this, number of rings are formed for every year. So this autumn season and spring season considered as a 1 year. 1 year. Like this, while the plant is growing by year, these rings also increasing. The same pattern. Same pattern means spring season, large, autumn season, very less. Now these rings called as the annual rings. Based on the annual rings, we can determine the age of the tree. This knowledge called as dendrochronology. This knowledge called as dendrochronology. Is it clear first? Annual
Yes,
rings
sir.
formation clear?
Yes, sir.
Right, but I have drawn only xylem set, but not phloem. You can assume the phloem also same way. Secondary phloem in spring season, highest number, less number secondary phloem, very less in autumn season. That's it. Is it clear?
Yes, sir. In 1 year, two rings will be formed, sir.
Right, two rings formed in 1 year. But this is clearly appeared in temperate region only, because the temperate region clearly follow the seasonal spring season, autumn season clearly. And right, spring season, early wood, autumn wood, right. They all completed. So dendrochronology, study of the annual rings. Now the formation of heartwood and sapwood. Anyway, after formation, secondary xylem, secondary phloem, while gradually increasing, what happened? For years and years, later, for example, let's draw another diagram here. So this is one sort of circle, the second circle, like this. While the secondary here, for example, this is secondary xylem. This is the next year secondary xylem. This is next year secondary xylem. What happened while the secondary xylem formation? One secondary xylem replaced the another secondary xylem. While the secondary xylem increasing, this primarily formed secondary xylem crusted in the center. The crust in the center and form as a dark color structure in center, dark color structure. That is called heartwood, and surrounding wood called as a sapwood. Let's see diagrammatically. What is diagram? So diagram not given. So, but I will explain here later. I will show the diagram. In older trees, the greater part of secondary xylem is dark. Greater xylem, secondary xylem is dark brown due to the deposition of organic compounds like tannin, resins and gums, aromatic substances, oils. So it is settled at central. Already I have seen, I have shown the just now the diagram, central innermost layer of the stem. This substance make it hard and durable, resistant to the attacks of the microorganisms and insect. This region comprises the dead elements because all elements, all secondary xylem elements are dead and crushed and which are highly lignified walls. Already we discussed in the secondary xylem walls. Secondary xylem, secondary xylem walls in chemicals mainly secondary wall made up of lignin, right? So the after in center that mean the water formed a secondary xylem. The secondary xylem crushed in the center and form as a heartwood. Heartwood. And peripheral side, that is light color. This is dark color, and outer side light color. The peripheral region of secondary xylem is lighter in color, known as the. It is actually sapwood. It is involved in the conduction of water and minerals. Waters and minerals. But the heartwood does not conduct any water and minerals because it's completely dead tissue and crushed in center. And it has the more durable due to the composition of the lignin. I will show diagram later about the sapwood and heartwood. Just comparison here. Heartwood duramin, another name, sapwood alburnum. Heartwood is the central part of the secondary xylem, peripheral part of the secondary xylem. So both of the secondary xylem, but the where the centrally crusted secondary xylem that is developed as a heartwood, and outside of the this crusted secondary xylem that is called peripheral secondary xylem that is sapwood. Due to the crush in center, it is turns into dark in color, and the outer one there is no crush. They clearly form the vessel cells. This is lighter in color. And the first one, deposition organic compounds like tannins are deposited, resins or gums, aromatic substances. It is commercially valuable due to this all composition of chemical. In organic matter, there is no deposition of organic matter unless the vessels, the resistance to the attack of microorganisms and insect. There is no resistance here. The compressed dead elements with the highly lignified walls. Walls are not highly lignified. Of course, lignified present, but not highly lignified, which provide mechanical support and here conduction of water and minerals from the roots to the leaf. Now this is inside the stem. Now this is all formation intrastem. Now let's see the extrastellar changes. In extrastellar changes, already told you, while the secondary tissues are formed inside and outside, in the steel, the pressure, exert pressure falls on the epidermis. Due to the pressure, the another meristematic need. This meristematic development from the cortex cells. So as the stem continues increase in girth, due to the activity of vascular cambium, the outer corticals and epidermal layers get broken and need to be replaced to provide new protect cell, right? So hence the another meristematic tissues formed that is called cork cambium or phellogen develop usually in the cortex region. That mean actually already you knew this inside the cortex, inside the steel changes after formation ring, cambial ring. For example, this is the inside. Both sides secondary tissues are formed. Secondary tissues, secondary brain, secondary tissues are formed. Due to the secondary tissues increasing, what happened? And also the formation of secondary tissues, where is high, where is low. Already we discussed. During spring season, rich amount of xylem formed. During autumn season, low amount of formed. So anyway, while forming like this for every year, the secondary tissues gradually push toward the center and destroyed in center. This is formed as a heartwood and surrounded by sapwood like this. And while it is increasing, automatically the exit pressure falls on the epidermis and cortex cell. This epidermis and cortex cells, right? So this is cortex region. In cortex region, actually these are the cortex cell. This some of the cortex cell can change into meristematic cells. Let's change color. Meristematic cells, and these meristematic cells form together combinedly, form as a ring. Form as a ring. This is called as a car cambium. This is called as a car cambium. Inside the steel vascular cambium, this one. Outside the steel, this is car cambium. This is called car cambium. So what happened after formation car cambium, or another name is phellogen? Now tell me what happened after formation this cambium ring in cortex? What happened after cambium formation inside the steel? Hello, hello.
Sir,
After formation cambium ring inside the steel, what happened?
it gives a pressure on the Cambrium.
But I am asking, during secondary growth, inside the steel, intra interfascicular cambium both together form as a ring, cambium ring, right? After formation cambium ring, due to the activity of cambium ring, what happened?
Girth will be,
Will what gain?
the thickness will be,
How the thickness increase?
stem, thickness increased
Right? How the stem thickness increase? What is the reason to increasing the stem thickness?
because of the secondary growth, your car Cambrium formation.
What formation?
Cambrium, both Cambrium rings formed, no sir, here.
Okay, cambium rings formed. From cambium ring, what are formed outside and inside? Due to activity of cambium ring, what are formed?
Carp formed.
I am asking about inside the steel. Inside the steel, inside the steel, before secondary growth, what happened? Actually, inside the steel vascular bundles, in vascular bundle, intrafascicular cambium,
And
between vascular bundle, interfascicular, both form as in a ring, right?
yes
Cambium
sir,
ring.
yes sir.
Now after formation, this cambium ring shows the activity. What type of activity that shown? What happened after formation cambium ring inside the steel? You said the thickness increase. That is right. But how the thickness increase?
Secondary xylem and phloem.
That's it. Increasing the secondary tissues.
Yes sir.
Secondary tissues inside the secondary xylem, outside the secondary phloem. Now tell me, after formation this cambium, what happened?
Secondary tissues will be
Right, same. Here also same. Secondary tissues are formed inside and outside.
yes
Let's
sir,
see what are the secondary tissues in cortex formed inside and outside of the cortex cell. Let's see. Is it clear first?
yes sir, yes sir.
Right. Now the only I have drawing here. I am drawing here only the phellogen or car cambium, but not steel cambium. Scar cambium. Anyway, from the cortex cell, car cambium formed. This car cambium or phellogen cuts off the cells of both sides. Just now we discussed. The outer cells differentiated into cark or phellum, while inner side secondary cortex. Simple. Green color here. Inner side what is formed?
Secondary cortex.
Right, that's it. SC, secondary cortex. Inside secondary cortex. Outside cork cells. Outside called cork or phellum. Cork or phellum formed. Actually, these outside cells are completely dead cells. So the cork is impervious to water due to the suberin deposition. In cork, suberin deposited, it is impervious to water in the cell wall. The cells of the secondary cortex are parenchymatous, which is living, outside non-living. Now the phellogen, this ring, phellogen and phellum, phelloderm, phellogen, phellum, phelloderm. Actually, this total not given wrong. Just with something. So anyway, all together form as a periderm. The secondary xylem and secondary phloem are formed inside the stele, and same time exert pressure on the epidermis. The meanwhile secondary protective layer periderm is formed. This periderm formed by the secondary growth in cortex by the formation of meristematic layers in middle of the cortex. This is called phellogen or cork cambium. After formation, this phellogen cork cambium, due to the divisions inside secondary cortex formed, outside cork phellum formed, and the tissues produced to the inside called secondary cortex or phelloderm. Secondary cortex or phelloderm. The phellogen, phellum, phelloderm all together form as a periderm. That means outside cork, right? This total structure, called as phelloderm. Sorry, periderm. Periderm. Form as a periderm. Actually, not given here. Formed as a periderm. So phellogen, this is the cork cambium. This one. This one. Phellum, cork cells outside. And phelloderm. Phelloderm means secondary tissues, secondary cortex. Secondary cortex. Now these three, phellogen, phellum, phelloderm. Total three called as a, together form as a periderm. Periderm. Is it clear? Hello?
Yes, sir.
Yes, sir.
Right. Periderm and like a bark. It is formed as a bark. I will tell you what is clear about the bark. Right. So this periderm part formed later. Actually, remember, write down here the periderm. Phelloderm different, periderm different. Phellogen, phellum, periderm, phelloderm, periderm. So due to the activity of the cork cambium, the pressures build up on the remaining layers, peripheral phellogen, and ultimately these layers die. Final outside the cells would be die, and it is called as a phellum. Now the bark refers to the all tissues exterior to the vascular cambium, include the secondary phloem. Now what is bark? What is bark? The simple. This side the actually a steel vascular cambium. Steel vascular cambium. Steel vascular cambium, what happened? Inside secondary xylem, right? Hello?
Yes,
What
sir.
happened outside secondary
Oh,
phloem? This is also common. Steel and outside the steel, what happened? Outside the steel. Are you able to see the marker? Hello?
yes, sir.
Yes.
Right, right. So outside the steel, the same, take a cambium ring in cortex. Cortex cell altogether form as a cambium ring, red color ring. This is cambium ring. Cambium ring. Right after formation cambium ring, what happened due to activity of cambium ring? Inside what formed, outside what formed?
Inside secondary xylem, outside
Right.
secondary.
Not secondary xylem. Inside. Not secondary xylem. Secondary cortex.
Ah, yes, sir.
Secondary cortex are formed inside. Outside cork cells. Cork. Cork cells. Actually, here no epidermis. For our understanding, I have drawn epidermis, but they no they already the epidermis ruptured. While the increasing secondary tissue, the epidermis ruptured and it replaced by the
Bark.
secondary tissues, right? Bark. Right. So cork cells. This is cork cells. Right. The bark is from secondary phloem from this region. Secondary phloem towards cork cell. This entire portion called as bark. Now tell me what are present inside the bark? Cork cell present,
Secondary cortex,
cork
secondary.
cambium present, secondary cortex present, secondary phloem also present. Is it clear? Clear about bark?
Yes, sir.
So all the tissue outside the vascular cambium, outside the vascular cambium. This is vascular cambium, right? This is vascular cambium. And outside the vascular cambium means from the secondary phloem to the cortex, sorry, cork cells. So they are all called as bark. It includes secondary phloem, periderm. The simple one, what periderm? Periderm. Already told you. What is the periderm? What is periderm?
Phyllogen plus phellum plus phylloderm.
Right. These three together form as a phylloderm. Is it clear? Any doubt in this?
No, sir.
Right. About lenticels here, the another important topic here, the lenticels also given. I think in our book, lenticels given or not? Do you have book? Hello?
Sir,
Second growth. Lenticels. Uh, what are the topics given in second growth? Right, lenticels also given. Bark, ring bark. But here the type of phylloderm, phyllogen, secondary cortex also right. If the bark, so already the formation bark clear. But it is two types of bark given in our book, page number 451, ring bark and scale bark. First ring bark, the cork cambium develops complete ring. Complete ring. You can already must have seen in our daily life. Where is ring bark? Where is scale bark? The name itself given ring bark, it is the complete ring. When you remove the bark, it is separated as a ring without break. and scale break. In many tropical trees, the strips of cork cambium overlapping one another. So they produce bark in the form of strips. Such bark known as scales. Example eucalyptus. You must have seen eucalyptus and psidium plant, jama tree. We can observe clearly. Can you can you can you separate the complete bark from this both tree? Whenever you observe the eucalyptus tree, like a scales, the scales are separated. Hello, hello.
Yes, sir.
For example, do you know neem tree?
Yes, sir.
Yes,
Neem
sir.
tree. From neem tree, you can cut the bark and separate. You can clearly peel it off. Complete ring. Whenever you try in jama tree or eucalyptus tree, can you peel it off the bark in both plant?
No, sir.
Try once again. What happened in jama tree? The actually the bark is very thin, like a
Uh,
paper, like a
yes, yes,
paper. It is
yes.
easily peel it off like a layers, but not rings. So that is called scale bark. And another the importance of this bark commercially it is involved in bottle cork is made up of the one of the special tree that is Quercus suber. It is um, it is called as bottle cork. This bottle cork made up of the bark of oak. The first phyllogen in oak arise in the epidermis. Already you know how the phyllogen form, right? When the tree is 20 years old, the first bark is removed. Why it is takes 20 years old? Why it is take 10 years old? Why the years it need to formation this type of commercial bark? Why the years are needed?
To know the quality,
Already are given the first bark is removed. That is first formed bark is removed. It is commercially no valid. And another and it is replaced by another new cork. Why these corks are formed? How these cork are formed by year and year? That means here the years are taking for production of secondary growth. Actually, can you assume the radial growth in from beginning of the plant? Can you say any plant from the starting of life, instead of primary growth shows the secondary growth in any one plant?
in
Hello?
dicards, it shows no, sir, secondary growth.
But my question is in dicards, can you see only regular radial growth instead of primary growth only can you see the radial growth or secondary growth?
Oh, sir. First primary growth on, then
Right,
afterwards.
first primary. That means here in these plants also it takes 20 years, almost 10 to 20 years time. In this only actually first 5 to 10 years shows the primary growth. The later the 10 to 20 years it shows the secondary growth. During secondary growth one bark is formed, but it is commercially not value. Why? And again secondary growth takes place, and again another bark is formed. This secondary bark is formed by the secondary phylogen. The second phylogen arises in the deeper layer of the cortex. Same which is also produce the cork. After 9 or 10 years, the second cork is stripped off. By this time, the substance thick and has the commercial value and has better quality than the first cork. The first one called virgin cork. The virgin cork also peeled it off. There is no commercial value. After 10 years, again, same phylogen formed in deeper cells of the cortex. And the same secondary tissue is formed inside and outside. As a result, same cork formed. Now the second cork has the commercially value. Now this cork is removed at intervals of 9 to 10 years. The cork oak yield up to 150 years. So this good cork is light and impervious to air. So the dots seen in the cork are lenticel. I will explain now the lenticels, where the lenticels are present. So in examination point of view, there is a chance to ask the short question about bark. If they ask you write bark formation and what is bark, types of bark, and commercial bark also. So commercial bark formed by who? Pircus suber plant. Remember the plant name. And another importance of bark, the bark of cinchona, which gives the quinine, and Cinnamomum geylanicum is used to control of malaria. Cinchona, quinine, bark of cinchona, which gives the quinine, and Cinnamomum geylanicum, it is used to control of malaria. The bark of Acacia arabica, Acacia, used in tanning process. So these are the commercial importance of the bark, right? Right. Now lenticels. Now lenticels. The, the certain region of phyllogen cuts off closely arranged parenchyma cells on outer side instead of cork cells. Actually, what are formed outside from the cork cambium? Hello? From cork cambium, which cells are formed outside? Cork cells are formed. Apart from the cork cells, now when their parenchyma cells also formed somewhere instead of the cork cells. These parenchyma cells soon rupture the epidermis, forming the lens-shaped opening cells called lenticels. These lenticels actually permit the exchange of gases, but not cork cells. But cork cells not permit the gases and water. So the exchange of gases takes place from the lenticels. So between the outer atmosphere, the internal tissues of the stem. This occurs most woody trees. So this is the lenticel structure. Let's see here. Uh, so somewhere here, this is cork cambium actually. The red color cork cambium. Are you able to see this diagram? Hello?
Yes,
Hello?
sir.
Right. The what's also clear? Cork cambium, secondary cortex, what's clear or not? If it is not clear, write down. The red color, what I drawn here, the red color, that is the cork cambium. Right? Inside secondary cortex, this part, secondary cortex, this one, secondary cortex. Actually, what is formed outside? Cork cells, right? Hello?
Yes, yes, sir.
Dead cork cells. Apart from the gut, somewhere, what happened? Instead of cork cells, the living cells, living parenchyma cells are formed like this. The living parenchyma cells are formed. These cells called as complementary cells. These cells formed called as complementary cells. When the complementary cells formed, this epidermis ruptured and it make a pores, some small pores. Through the pores, gases exchange takes place. This is about the lenticels. Is it clear?
Yes, sir.
So anyway, lenticels, right. Any other extra matter in our book? about lentil cells. What will give it? Lentil cell by formation periodium stomata, closed stomata. Okay, lentil cells. Right. So this is lentil cells act as a like a stomata for exchange of gases. In leaf, exchange of gases through stomata, but in tender stem also stomata there. But in secondary growth, after bark formation, also now and there some of the complementary made of cells are there. So this is for exchange of gases to internal parts. It's clear? Hello?
Yes, sir.
Yes, sir.
Right. Secondary growth in roots, no need. Only stem we require. Right. Now, any doubt in secondary growth? Any doubt in secondary growth? Hello? Any
No,
doubt
sir.
in intrastellar? Any doubt in extrastellar? Any doubt formation, phylogen, phellum, cork, periderm? Clear?
Clear, sir.
Right.
Yes, sir.
Now let's see the anomalous secondary growth. Anomalous secondary growth. Not here. Right. Are able to see anomalous secondary growth slide? Hello?
Yes, sir.
Right. What is anomalous secondary growth? This is also important for long question. Already you must have learned during practical training classes. Anomalous secondary growth in Bignonia, anomalous secondary growth in Amaranthus, in Dracena, right?
Not explained anything, sir.
Okay, no, don't worry, I will explain again. Right. So first of all, what is anomalous secondary growth? Just a rough definition. What do you think yourself? Just I want the definition. I want your
Anomalous.
concept,
Maybe it is a reverse process for the secondary growth.
not reverse process.
Mm.
Somewhat right. Reverse to the secondary growth, but but not activity. That
That
means
that is only, sir.
actually right.
Not general process to the secondary growth. It
Right.
is
Exactly. So what happens in secondary growth? Generally, cambium ring forms inside the steel and secondary xylem phloem formed in a normal way. That is normal secondary growth. But any changes in the normal secondary growth, any changes, that is called as anomalous or abnormal secondary growth. The any changes may be in our book clearly given. The normal, the primary structure in dicot shows following character. Already you know the primary characters. Don't discuss about that. Anomalous primary structures here given. What are the anomalous primary structures? Occurrence of the scattered vascular bundle in dicotyledon. Is it normal? In dicotyledon, how the vascular bundles are arranged? Hello? Just now we discussed the vascular bundle arrangement. The vascular bundle arrangement scattered manner or a ring manner?
Ring manner.
Ring manner, ring. But what is the anomalous activity? Here, the clearly given 19.4.1, the occurrence of scattered vascular bundle in dicotyledons. But how the vascular bundles in dicotyledon? Actually, the vascular bundle in dicotyledon has a ring. But instead of that, they are scattered manner like a monocot. That's why it is the anomalous character. Presence of medullary bundles. Where are the vascular bundles? Hello? Inside medulla or outside medulla? Hello? Hello? Are you follow me? Where are the bundles? Where are the bundles? Inside or outside? In general, I am asking in general. Bundles, vascular bundle. Ayo. Just now we completed about the secondary growth. This is the steel vascular bundle in a ring outside the medulla, but not inside. But the anomalous character is presence of bundles in medulla and presence of this bundle same in cortex. In normal way, vascular bundle present in cortex. Hello, hello.
No, sir. No,
My
sir.
voice is clear, right?
Yes,
So
sir.
absence of vessels in the xylem. That is also abnormal character. If you take the secondary xylem, it should consist the vessels. If the vessels are absent, that is also anomalous character, abnormal character. The presence of exclusive phloem and xylem bundles, exclusively phloem in xylem bundles. Actually, in general, xylem more than phloem. But if this condition reverse, that is abnormal. And polystelic condition, actually one stele is there. If the more than one, that is polystelic condition, that also abnormal condition. And occurrence of internal vascular bundle. Internal vascular bundle, they are all abnormal character. First, let's see the first one given scattered vascular bundle. Scattered vascular bundle means actually in dicots, you know already the dicots, the vascular bundles are in ring. But the vascular bundles are not arranged in ring but scattered in the pith medulla. This condition found in the stem of family. Given example Nymphaeaceae, Nymphia, Pepaveraceae. So these are the examples, right? And where the medullary vascular bundle present, I will explain one by one later. But medullary vascular bundle, already medullary vascular bundle, you have taken section cutting. Medullary vascular bundle, Boheravia, best example Boheravia. So cortical bundles. So bundles are present in cortex. The vascular bundle present in cortex. Example Casuarina. The vessels absent in xylem. Utricularia. Example Utricularia. Exclusive phloem, xylem bundles. Example: Acranthus aspera, Mirabilis jalapa. Polystelic condition. The polystelic conditions also Primulaceae family. Internal vascular bundle, found in internal vascular bundles also some other plants. During the normal secondary growth, the secondary tissues are formed in an unusual method. That is abnormal condition. So if you see the unusual condition, that is anomalous secondary growth, takes place by under following methods. Normal cambium with abnormal activity, abnormal cambium with abnormal activity, formation of accessory cambial rings, formation of extrastellar rings, formation intraxillary phloem, formation of interaxillary cork. So let's discuss one by one. First of all, normal cambium with abnormal activity. So abnormal activity, here the example, Aristolochia. Monocot stem, Aristolochia. Actually, Aristolochia not given. Okay. So anyway, I will explain this normal cambium with abnormal activity. In many woody climbers or lions, the cambium is in normal position, but it will behave in an abnormal way. As a result of this, anomalous structures are formed. The stem, which is another one is Aristolochia. I think Bignoni also, but not given. But actually, instead of Bignonia, in our plants given Aristolochia. Yes, Bignonia also given. Second. Remember, in 4.457, one of the abnormal character, that is the normal cambium with abnormal activity. The normal cambium ring with abnormal activity, two plants given example, one is Aristolochia, another with Bignonia. Already Bignonia you must have learnt in practical training classes, and again I will explain.
Yes,
So
sir.
right, what is here the simple title is heading is normal cambium ring. Just draw. Let me draw cambium ring. This is normal cambium ring, and it shows the abnormal activity. That is important. This normal ring shows abnormal activity. Right. Let's see how it shows abnormal activity, especially which is found in lions. Lions. I will explain. So in general, tell me the secondary xylem formation inside, secondary phloem formation outside, which is abundantly formed, xylem or phloem.
Secondary xylem fluid open.
Right,
Xylem
secondary xylem. Right. Secondary xylem formed abundantly, then secondary phloem. That is normal activity. But what happened in this? What happened in this? Instead of this reaction would be reverse. Means actually somewhere here.
secondary phloem.
Yes.
In Bignonia, four sides, four patches will be formed, no, sir?
Yes,
Right.
yes. Actually, right. Actually, what happened in general, cambium ring with abnormal activity. Uh, Out secondary it is. right outside secondary phloem is completely secondary phloem. Secondary xylem. Here secondary xylem. Right. This is normal condition. But what happened due to the abnormal activity? It not uniform. So somewhere instead of formation high number of abundant abundant of auxiliary elements, here form just very less amount auxiliary form. Here also same less amount auxiliary form. Here common. Here less amount auxiliary form. Now let's change color. At this region completely filled by secondary phloem. Here also secondary phloem. Here also secondary phloem. Let's take another one. This one also take. Right. The red color common normal activity. No problem with this. Because in normal way, secondary xylem abundantly formed, then secondary phloem. If you see this region here, here secondary xylem high, then secondary phloem. Let's change color. Secondary phloem. This region, secondary phloem, very less. This region. But due to abnormal activity now and there, what happened here? The secondary xylem, very less amount here. We can observe here, and replace the rest of the it is replaced by this completely formation as a secondary phloem. Secondary phloem. Is this abnormal activity or not?
Abnormal.
Right? Is this abnormal activity? Like this abnormal activity occurs in Bignonia family. One example is enough. Bignonia family. One example is enough. The anomalous secondary growth structure Bignoniaceae lions. It is an adaptation to their habit. What is lion first? What is lion? Hello?
Sir,
What is lion? Lion means woody stem like a twinner nature. Woody stem, it shows twinner nature. That is called lion. So actually, as it woody nature, strong nature, can it bend easily? Hello? Can it woody nature, can it bend easily? Simple. Simple here. For example, let me draw another one. This completely secondary xylem. Complete secondary xylem. Already, what is the function of secondary xylem? Hello? Function of xylem. Secondary xylem function. And outside the secondary phloem. This secondary phloem. If the plant appear like this, presents like in this condition, the secondary xylem gives anchor to the stem, support to the stem. Hello, are you follow me?
Yes, sir. Yes.
It gives anchor to the stem. But lions externally appear as a strong, but it can easily bend due to the formation of high amount of secondary phloem instead of secondary xylem. If the secondary xylem present high, it is not possible to bend easily. Hello,
Sir.
that's why it is adapted. Adapted means due to the adapted the abnormal activity of cambium ring, the some places actually it is started with four ridges, four ridge like these are called ridges, four ridges. It is extended to the 8, 16, 32. That is completely extended. The ridges number extended, right? So due to the ridges, due to the composition of secondary phloem, this second does the secondary phloem give the anchor to the plant body? Hello? Does the secondary phloem gives anchor to the plant body? Does the secondary phloem gives the support to the plant?
In this
Hello?
situation, secondary phloem only, no, sir.
Yes, only phloem present, but does it gives the anchor to the stem? Does it give support? No, the secondary phloem does not give the support to the
How secondary
It
phloem
is not a
is.
mechanical. Only xylem is the mechanical.
Ah,
That's why it's bends easily. It bends easily, but externally it appear as a woody stem. Even though it is appear as a woody stem, it bends easily and takes support to grows upright direction. So that is the condition of lion. Is it clear? Hello? Hello?
sir, yes, sir.
Right. And another one is abnormal cambium and abnormal behavior. Actually, it is not in our practical training classes, but I will explain here. Abnormal, first one, normal cambium, abnormal activity. But second one, abnormal cambium with abnormal activity. So the abnormal cambium generally originate in the cortex or pericycle. Actually, in the cambium ring, generally originate in where? Hello?
Sir, Cambrium, sir.
Cambium. Vascular cambium actually generally developed in
Medullary region.
right steel region. But
Ah,
abnormal cambium generally originate in the cortex or pericycle. In those cases where either the normal cambium ring does not develop at all or ceases the functioning. So due to the absence of normal cambium in steel, instead of that, abnormally this cambium ring formed in cortex or from pericycle. Due to its unusual position, the function of cambium abnormal. Simple one word. I also given the one of the example here. Uh, Serraginia. Another example, Tinoia verticosa. These two examples. So first remember the formation of cambium ring wrong. That is abnormal. Abnormal ring formation. Where the abnormal ring formed abnormally in the location of cortex or pericycle. That's it. And activity of a different. After formation, this ring, this activity, the activity of also the secondary tissues formed inside and outside abnormally formed. So during the secondary growth, all the cambium ring of vascular bundle functions independently and produce secondary xylem, inner side, and secondary phloem, outer side. Thus, the stem consists of several discrete vascular cylinder. So there is large central stele surrounded by few peripheral ones. Hence, each vascular cylinder finally develops into one periderm. Actually, all the vascular bundles combined together form as a periderm. But what happened here? Each vascular bundle individually develop periderm. That is abnormal activity. Hello?
Yes, sir.
So what I'm saying here,
Each vascular bundle forms a periderm.
right? That's it,
Hmm.
right? And formation of axillary cambial rings. The best example, Boeravia given in our book in this slide also. Just wait. This is Boeravia. Sorry, Dracena. This one Boeravia stem. Let's see what are the abnormal activity. In actually in any stem, first primary structure common. What the primary structure means, epidermis common, cortex common, pericycle common, vascular bundle. Here, this is abnormal activity. Already told you. What is abnormal activity in this? Let's see. Vascular bundles are present in 3 rings. This is abnormal. Actually, how many rings vascular bundle? Hello, vascular bundle. How many rings in general?
One ring.
1 ring, but here 3 rings. That is abnormal activity. The innermost ring present in the 2 large bundle in middle of the ring number 6 to 14 and outermost 15 to 20. So vascular bundle innermost, middle rings are medullary bundles. Vascular bundles are conjoint collateral endarc like a dicot stem. The two vascular bundle of the innermost rings are large, oval lie opposite each other. ఇదంతా ఎందుకు? This one. So this is medullary rings. Medullary rings. This is middle rings. This is the outer rings. But the secondary growth occurs in outer ring only. Secondary growth occurs in outer ring only. Right? This is another abnormal character in Boeravia stem. Remember, Boeravia 3 rings are present. The 1 ring, the present inside the medulla, medulla region rings. That is the abnormal character. Right?
The
So the
scattered vascular bundle also anomalous feature only, sir.
which one?
Scattered vascular bundles.
A scattered vascular bundle is abnormal. But scattered vascular bundle, if it is found in dicot,
Ah,
but not monocot. Monocot in general is scattered.
scattered.
In dicot stem, if any scattered bundle from that is abnormal character. So
Okay.
anyway, after formation like this, due to the abnormal activity, the outer layer, what happened is I will draw only outer vascular bundle. Outer vascular bundle forms the cambium ring, but this cambium ring not formed in inner and middle. After formation cambium ring, like internal secondary tissue formed inside and outside. After some time, after a few days, this cambium ring activity not shows. This activity does not continue. Now another ring formed, another cambium ring formed. Same, this also formed the secondary tissues inside outside. And after a few days, this also undergoes to dormant. And again another forms, another ring formed. Like this, concentric rings are formed in Boeravia. So this is also abnormal character. In Boeravia, first abnormal character position of vascular bundle in medulla, that is abnormal. In terms of activity, actually after formation vascular bundle, it shows secondary growth sometimes and again stop secondary growth and newly formed cambium ring. This is continuous every time. That way concentration rings are formed. This is in Boeravia. And another one is another abnormal secondary growth in found Dracena, that is best good example and a very important monocot stem. Actually, monocot stem, can you find out the secondary growth in monocot?
No, sir.
Hello? No,
No,
actually, if you take any monocot like rice or wheat, they are very thin, not they radially not grow. Only it shows vertical growth. Grasses family, they are all very thin. There are no secondary growth. Due to lack of secondary growth, it is very thin. But this secondary growth present in stem, if the secondary growth present in the stem, that would be the abnormal. or anonymous. Let's see the monocot stem example. Dracena. What happened in Dracena? This is epidermis, cortex. This is all common. So these are the cortex ground tissue. In ground tissue, vascular bundle present. Actually, these vascular bundles, first primary vascular bundle commonly found, next secondary vascular bundle formed from the cortex cells. That is abnormal character. Here the vascular bundle is actually not a concentric vascular bundle. We can see here. Secondary bundles are most lumpy vessel. Okay. Where it is. So it's a monocotyledons in regions have the seized elongates, no. Campbell cells fusiform rectangle, no. This formed side row, no. No furious cambium divide longitudinally, initially anticlinal, right. Xylem prime element, secondary xylem. These secondary bundles are mostly emphasized. Just wait 1 minute. One is these are actually vascular bundle. They are not conjoined collateral bundle. Xylem cells surrounded by and center phloem. This is one of the abnormal activity in monocot stem, first of all. But later, the secondary from the secondary tissues, again, the same vascular bundle formed with help of the conjunctive tissue. Actually, this conjunctive tissues also developed into meristematic cells, later formed as a center phloem and surrounded by xylem cells. Of course, they are scattered like a more normal monocot stem. But what happened in normal monocot stem? In normal monocot stem, the phloem xylem arrangement in a vascular bundle. This vascular bundle, phloem outside and xylem inside. This is the condition. But how the Dracena vascular bundles? In Dracena vascular bundles, concentric, not conjoint. Same radius, this is conjoint. But if you see the Dracena, concentric, central poem, but not outside, and surrounded by xylem cells. This condition is the abnormal activity. And due to the activity of conjunctive tissues, actually before formation, the xylem and phloem cells, the conjunctive tissues developed into cambium ring, cambium cells. Cambium cell, what happened after formation cambial ring? Hello? What happened after formation cambium ring? The cambium ring formation forms. What is the activity of cambial ring? Hello? Are you there? Hello?
Yes,
Yes,
sir.
sir.
Tell me what is the activity of cambial ring? Actually, first of all, remember in monocot no cambium ring. But however, the cambium ring formed from the ground tissue or conjunctive tissue. Then what happened after formation cambium ring? After formation cambium ring, secondary tissues are formed inside and outside, like phloem and xylem cells. After formation, the phloem and xylem cells, these are developed into concentric vascular bundle. Hello?
Yes, sir.
So this is the abnormal activity in monocot stem. So this is called secondary growth, abnormal secondary growth in stem. And rest of the we will complete next class. Rest of the secondary growth. Is it
Okay.
clear?
Yes, sir.
Right. Any doubt in secondary growth or abnormal secondary growth?
These all stems we have to learn, sir. Boiravia, Dracena, big one,
Yes,
all.
all are important. All are important.
Okay, sir.
Okay. Can I quit?
Okay, sir.